And today we are going to start next topic of the same chapter, okay, that is enzymes. So you can see here in front of you the complex molecule, right?
Yes, ma'am.
So there is something who can cut this complex molecule and make the uh, the substance into the simpler form. Okay, so can you guess what is that? Here amylase, sucrase, isomaltase, maltase, lactase, all these things can digest this complex molecule present in the bread into the simple sugar, glucose. What is that substance? These are the
Enzymes. Ma'am.
exactly enzymes, right? So enzymes have the ability to uh, catalyze the reactions, right? Enzymes have the ability to digest or convert the complex molecule into the simpler form, right? So today we are going to discuss about the enzymes, the structure, the uh, properties, the actions, mode of actions, okay, the nomenclature types and all that. Okay, in the previous session, you must have learned about the biomolecules, right?
Yes, ma'am.
Different types of biomolecules. Can you explain me what are the different types of biomolecules? Yes.
Ma'am, protein
Okay.
and hydrogen,
Hydrogen.
carbon,
Hmm.
phosphorus.
So these are the inorganic molecules, the ions. Okay. Apart from that, the biomolecules, the molecules, macromolecules, micromolecules, right? So macromolecules, give me the example of macromolecules. Large molecules, carbohydrate, lipid, protein, right? Nucleic acid. So here I'm taking one of the example, protein. Okay. In that you must have learned about the structure of protein, the properties of protein, characteristic of protein, uses of protein, classification of protein, right?
Yes, ma'am.
So let's say the structure of protein. Okay. So in protein, the structural organization, there are four types of structural organization: primary structure, secondary structure, tertiary, and quaternary. The secondary is again, there are two types, alpha helix and beta pleated sheet. Okay, this is beta pleated sheet. Okay.
Yes, ma'am.
Can you explain me the difference between alpha helix and beta pleated sheet? On what basis you call it alpha helix or you will call it as a beta pleated sheet?
Uh, the arrangements of biomolecules, ma'am.
Arrangement of biomolecules. But here we are talking about the protein. Only protein, not the whole biomolecules. Hmm. Yes. Okay, tell me one thing. After each session, what you used to do? After each session? Yes, Aarti.
Yes, ma'am. I do class assessment, ma'am,
Uh,
and eat and sleep, ma'am.
Eat and sleep. Okay. And in one session, how much topic we used to cover? The whole chapter? No.
No, ma'am.
Only the small section of the chapter, right?
Yes,
So
ma'am.
it's common, it's normal to expect that you will read only that particular segment what we have completed in the session, and that one you are not doing. So if parallelly you are not revising, then how I will expect that you will do at the end? Because you know now the 11th syllabus is very vast, right? The topics are very new and it is vast also. And the weightage is very high in NEET. How you will cover all these things? Make a habit, okay, to revise particular topic what you did in the session just after the session. At least do the simple reading. No need to mug up the definition, write all the concepts. No, don't do that. Just read that, because just now you have finished to understand the concept. And after that, once you will read all these things, everything will go and stick inside your mind. Okay? And this is very simple way. No need to go after the finishing of chapter and do the revision, okay, spatially. No, don't do that. Okay? So from next
Yes,
time,
ma'am.
if I will ask you the question from the previous session, I want answer.
Yes, ma'am.
Okay?
Yes, ma'am.
Pakka?
Yes, ma'am.
Okay, let's see. If you will not do that, I will not teach. Okay? Chalo. So alpha helix is the right-handed screw or the helical structure. Okay? The main difference is in pleated sheet, the polypeptide chain is held by the side-by-side hydrogen bond. Okay, whereas in helix, the polypeptide chain is held by the hydrogen bond in between. This is the main difference. Are you getting me? Here, hydrogen bond is present internally. Here, the hydrogen bond is present side by side. That's it. Okay? Chalo. Now let's start our next topic, that is enzyme. So enzymes are the biocatalyst. Biocatalyst. Bio means bio means
Living, ma'am.
living, and catalyst means
Um, in reaction, catalyst increase the speed, ma'am.
okay. So any substance which increases or accelerate the reaction is catalyst. Okay. So enzyme is is the catalyst which increases or enhances the reaction, chemical reaction inside the cell, inside the living body. So it is living catalyst or biocatalyst. Okay, enzymes are made up of protein. Only one enzyme which is non-proteinaceous, and that is ribozyme. Ribozyme is made up of RNA, that is nucleic acid. Okay, except ribozyme, all the types of enzymes are proteinaceous. But it does not mean that all the proteins are enzymes. No, all the enzymes are protein, but not all the proteins are enzymes. Are you getting me?
Yes, ma'am.
Okay. So when enzyme is accelerating the chemical reaction inside the cell, they accelerate the reaction. They either reduce or enhances the activity, okay, by the activation of reactant, the substance, the energy, but it will not affect the end product, the final product. Okay, it will it will just enhance the reaction. It is just a supportive system, but it does not affect the property, nature, or the structure of final product. Clear?
Yes, ma'am.
So this enzyme was first time discovered by the Butcher. Okay, this Butcher was the German biochemist and he discovered enzyme in yeast. Okay, and he said that something is there in the yeast cell that enhances the activity inside the yeast cell, metabolic activity, or the cell is active or living because of that particular substance. But what is that substance? So Wilhelm Kuhne, he coined the term that the substance what the butcher have discovered in the yeast was nothing but the enzyme. Okay, he coined the term enzyme. Okay, and then another scientist came, that was J. Sumner. He isolated the enzyme from the bean. Which bean? Jack bean. Okay, he isolated the enzyme from the jack bean. And which enzyme? Urease. Okay, and then he crystallized that, the pure form of enzyme. Okay, so this is the history, and out of which the most important one is Wilhelm Kuhne. Okay, now the properties of enzyme. Here we are having the list of physical property like biocatalyst. What is the meaning of biocatalyst?
Which increase the speed and do not
Catalyzes,
affect,
it
do not
catalyzes the biological reactions, right, in the living one. So it is
like
biocatalyst.
catalyst.
Okay. Another property is precipitation. What is the meaning of precipitation? Precipitation.
Precipitate, ma'am.
Sorry.
Um,
Yes.
the undissolved, ma'am.
Undissolved.
Dissolved, not undissolved, ma'am.
Yes.
The product which cannot dissolve in water, ma'am.
That is precipitation. Sure. Hmm. Enzyme precipitation. Okay, so whatever acidic solution or alkaline solution, okay, once you mix with the enzyme, it will make the enzyme precipitation. Okay, precipitation means get condensed and and what is the literal meaning of precipitation? Hmm.
Which settle down on the water, ma'am.
Settle down in the water. Can we say that the liquid form or frozen water? Huh? Then precipitation means what? Rainfall is what? Precipitation? Precipitation? Precipitation of what? Water vapor, right? First the evaporation will happen, then condensation of water vapor, and then precipitation will happen, right? So, and precipitation is what? Rainfall. So precipitation is any liquid or frozen water. Okay. So if it is in the form of rainfall, so here precipitation in enzyme. So enzyme, we already know it is a liquid, right? The fluid substance. So once you will add alkaline or acidic solution to the enzyme, enzyme precipitation will take place, which means, which means, which means, which means, am I audible, Aarti? Hello?
Yes, ma'am.
Yeah, which means that if the solution if the enzymes are amphoteric. Okay, amphoteric means the amino acid or amino acid side or amino end of the amino acid and carboxylic group end of the amino acid in the chain. Okay, that is called amphoteric. Okay, so enzymes are proteinaceous in nature. We already know, and protein is made up of amino acids. So enzymes are amphoteric. Okay. So when you add the solution acidic or alkaline, it will precipitate, means it will neutralize or it will make it more watery. Are you getting me? So this is enzyme precipitation. Molecular activity. What is that molecular activity?
Um, inside the molecule, the activities.
Yes, suppose you are having carbohydrate starch. Okay, starch. If you will add amylase, if you will add amylase, what will happen? The molecular structure of starch, it is a polymer of glucose, right? So it will break the molecular structure and make it glucose, right? So here the enzyme is acting on the molecular structure. It is showing the molecular activity. Are you getting me? It does not changing the conformation or it is not changing the morphology. Are you getting me?
Yes, ma'am.
Enzyme activity. What is that enzyme activity? The catalysis, the reaction, enhances the reaction. Okay. Denaturation. If enzyme is under the unsuitable condition, enzyme can work properly only under the optimum temperature, optimum pH, optimum substrate concentration. Okay, but if the enzyme comes in a contact with any adverse condition, high temperature, then what will happen? Enzyme will denature. Denature means the activity will modify. Now the enzyme will not perform the function, the catalysis function. Are you getting me? Solubility. Yes, enzymes are soluble. If you will mix with any protein, any biomolecule, okay, the water, it will easily mixed. Colloids. What is colloid?
The mixture, ma'am.
Mixture. Okay. So enzyme behaves as a colloid because of their large size and high molecular weight. Okay. Because of large size and high molecular weight. Okay. So
Yes.
just because of their large size and molecular weight, enzymes will start behaving like a colloid. Okay. Clear?
Yes, ma'am.
Now let's see another property. What happened, Arthi?
Ma'am,
Yeah. Are you getting
nothing.
what I'm trying to explain you is clear to
Yes,
you? Are you
ma'am.
getting the concept?
Yes,
Or you
ma'am.
want me to explain the things again?
No, ma'am. Clear only.
Okay. Now let's see another thing. As we already discussed that all the enzymes are proteinaceous, are made up of protein, but all the proteins are not enzymes,
Yes,
right? Enzyme is catalysis, biocatalyst, because it accelerates the reaction without undergoing any changes in them. Without changing itself, enzyme will accelerate the reaction. It will affect the rate of reaction without affecting themselves as well as without affecting the end product. Okay.
yes, ma'am.
Enzymes are very sensitive to pH and temperature especially. Okay. Enzymes are very sensitive to the pH as well as temperature. Okay. They are very specific, pH specific. They are very substrate specific. They are very temperature specific. The optimum temperature for enzymatic action is 37 degree Celsius. This is the optimum temperature. Sorry, yeah. Okay. If the enzyme is comes in a contact with higher temperature, more than 37 degrees Celsius or less than 37 degrees Celsius, then what will happen? If the temperature is around 0 degrees Celsius, at that time enzymes are inactive. Here the enzymes are inactive. Okay, and here the enzymes will denature. Are you getting me? Enzymes will work properly only at the optimum temperature, that is 37 degrees Celsius. Okay, our normal internal body temperature is also 37 degrees Celsius, and that's why all the enzymes present inside the cell perform their metabolic reactions or accelerate the metabolic reactions. Clear?
Yes, ma'am. Ma'am, denatured means inactive only, no, ma'am.
No, denatured means they have lost the enzymatic activity. Inactive means the structure will, the ability will be remain same, but right now they are not performing any function. Denatured means never
They
they,
lose.
yeah, they will lose the ability to react, to accelerate enzymatic activity. Okay, after the 60 degrees Celsius, once you will regain the normal temperature 37, that enzyme will not work because the enzyme have already lost the property. Okay, that is denature. Inactive
Yes, ma'am.
means right now not performing, but once it will get the optimum temperature, it will perform. Clear?
Yes, ma'am.
Okay. Now let's talk about pH. So the optimum pH for the enzymatic activity is 7. Okay. Optimum pH is 7. Okay.
Yes, ma'am.
But there are some what we call enzymes who perform their activity even below the 7 and even more than the 7. For example, pepsine or renin. You must have learned in your previous academic year the digestion, right? At that
Yes,
time,
ma'am.
the gastric gland secretes what? Pepsinogen. That is inactive form of enzyme, right? As well as it secretes HCl, which is acidic, right? Acid. So this acid creates the medium, the environment acidic. So once the pepsinogen enters into the acidic medium, because of the acidic condition, that is below the 7, near about 2, okay, the enzyme gets activate and turns from inactive state, that is pepsinogen, to the pepsin. Now the pepsin will work. Now the pepsin will digest the protein, right? So
Yes, ma'am.
there are some exceptions. So please write it down for the pH. Exceptions, pepsine and pepsine and renin. pH is 2. There are some enzymes who can work properly at pH more than alkaline phosphatase. Alkaline phosphatase. And it will work in a high basic medium, that is 10. Clear?
Yes, ma'am.
Okay. Now the uh, substrate. Yes, it is highly substrate or reaction specific. Okay, all the enzymes cannot work properly for all the substrate or for all the reaction. Okay, pepsin will work only on the protein and not on the any fat molecules. Are you getting me?
Yes, ma'am.
Yes. Enzymes also required very small in amount. Okay, in a very small quantity you can accelerate the reactions. Are you getting me? So enzymes are required in a very small quantity to accelerate the reaction. This is the another characteristic or property of enzyme. Okay, the catalytic power of enzyme is represented by the Michaelis-Menten constant. What is that Michaelis-Menten constant? This Michaelis-Menten constant is actually also called as Km constant or turnover number. Km constant or turnover number. As this constant was given by the Michaelis-Menten, so this Km constant is also known as Michaelis-Menten constant. Okay. So this Michaelis-Menten constant, how you will define it? The number of substrate molecule converted into product per unit time by one molecule of enzyme. Are you getting me?
Yes, ma'am.
This Km constant is for substrate. Substrate concentration. Okay. The number of, please write it down, the number of substrate molecules converted into product, number of substrate molecule converted into product per unit time by one molecule of enzyme. By one molecule of enzyme in favorable condition. Okay. Favorable condition means optimum temperature, optimum pH. Then only it will convert into the product. Okay.
Yes, ma'am.
Done, Aarti?
Yes, ma'am.
Okay. Now let's see the structure of enzyme. Okay. As per their structural composition, we already know that enzyme is what? Proteinaceous, means it is made up of protein.
Protein.
If any enzyme is made up of only protein, there is no additional metal ion or anything, okay, only and only protein is there, then this type of enzyme is called this type of enzyme is called simple enzyme. Okay, and the example is pepsin, trypsin, pepin. Okay, these are the simple enzyme because it is made up of purely protein. There is no additional thing. Are you getting me? The
Yes, ma'am.
polypeptide chain, the chain of amino acid only. Okay, another type of enzyme as per the structure is conjugated enzyme. Conjugated enzyme is also called as holo enzyme. Either it will be the apoenzyme or the cofactor. Okay, so this conjugated enzymes are made up of one or more polypeptide chain. Okay, one or more polypeptide chain. Okay, along with the protein, there are some non-protein substance, and that is cofactor. cofactor. Protein plus some of the non-protein component, that is cofactor. Okay, if it is bind with that, then it will be called as conjugated enzyme. So this cofactor once bind with the protein, make it enzyme, and it gives the enzymatic activity to that particular protein. Okay, and here in the conjugated enzyme, the protein part is called as apoenzyme. Suppose this is the protein structure. Okay, and some of the non-protein part, that is cofactor, is attached to that. Sorry. Okay, so this one, the whole together is called as conjugated enzyme or the holoenzyme. In this holoenzyme, the green part F is called, or the protein part is called apoenzyme. Apoenzyme, which is the protein part of the conjugated enzyme. Protein part. Whereas non-protein part is cofactor. Cofactor, that is non-protein part. Okay, now there are two types of cofactor. One is prosthetic group and another one is coenzyme. Okay, so basically there are three types of cofactor: coenzyme, prosthetic group, as well as the third one is metal ion. Okay, three types of cofactors are there. Okay, metal ion, coenzyme, and prosthetic group. So what is this metal ion? What is this metal ion? Sorry. Yeah, let's start with the metal ion first. So metal ion, actually the normal metal like iron, manganese, cobalt, zinc. Normal metal what you have studied in your chemistry. Okay, so these metals, when attached to the protein, okay, apoenzyme, and it regulates the activity of enzyme by forming the coordination bond. Please remember that. Forming coordination bond. Okay, this metal ions forming the coordination bond with the side chain at the active site. Side chain of active site. Okay, active site in protein, apoenzyme. Clear? And then it will regulate the enzymatic activity. Okay, for example, zinc. Okay, zinc activates the or enhances the metabolic reaction in photosynthesis. Okay, glycolysis. Now let's see the coenzyme. Coenzyme is the organic, non-protein organic compound which are loosely attached to the apoenzyme. Okay, these organic compounds are loosely bind with or attached to the apoenzyme. Whereas prosthetic group, which is again non-protein part, it is tightly attached to the apoenzyme. Okay. Coenzyme is also an organic group, and prosthetic is also an organic group or the organic compound. But the difference is, if it is loosely attached to the apoenzyme, then it will be coenzyme. If it is tightly attached to the apoenzyme, then it will be the prosthetic group. And why they are loosely attaching or tightly attaching? Because of their structural constraints. Okay. For example, coenzyme. Coenzymes are made up of vitamins, okay, like nicotinamide adenine dinucleotide, NAD, NADP, okay, NAD, NADP, nicotinamide dinucleotide phosphate, okay, FMN, FMN. In the next unit, the plant physiology, you are going through all these terms, okay, in the photosynthesis, in the phosphorylation process. Are you getting me? Okay, so these are the example of coenzyme. Prosthetic group, as I told you, it is the, or again, an organic compound which is tightly attached to the apoenzyme. Okay, and the examples are catalase, peroxidase. So the prosthetic group, okay, when attached to the protein and make the enzyme firmly that break down or digest the complex molecule into the simpler form. And the example of that type of enzyme is catalase, that will catalyze or cutting, okay, breaking down. Peroxidase. So peroxidase will breaking down the H2O2. It will break down the H2O2 into H2O plus oxygen, free oxygen. Okay. Structure is clear to you till now? So that was what? The molecular structure, okay, the protein and non-protein part. Now let's come to the morphological structure. So the structure, the whole structure, okay, is having two sites, active site and allosteric site. Active site is the specific part of the enzyme which is very specific to the substrate to bind. Okay, this is the site where the substrate will come and bind. So this site, the active site, will always be specific, okay, to the particular substrate. So that only specific type of substrate will come and bind to that particular land, that particular site. Clear? Till now it is clear to you? Okay. So once the specific type of substrate will come and bind to their related active site, after that, what will happen? The reaction will catalyze. Are you getting me? So what will be the definition of active site? Specific part of, write it down, specific part of enzyme at which specific part of enzyme at which specific substrate is to bind and catalyze the reaction is what? Active site. So this active site is what? Is the protein part. Okay, active site is the protein part which is made up of very specific type of amino acid, and this amino acid is actually coded by the genetic codes. Okay, Another site of enzyme is allosteric site. Allosteric site is the site which is besides the active site. Okay, where or you can say that this is the additional site in enzyme. Okay, where the other than the targeted substrate will bind. Okay, that will regulate either the reaction will takes place or not. For example, if any inhibitor will bind to that. Okay, if any inhibitor will bind to the additional binding site, then what will happen? The enzyme will not work because this inhibitor is inhibiting the activity of enzyme. Are you getting me? For example, lac operon. So in lac operon, suppose this is your uh, DNA. Okay, it is having the genetic sequence. Okay, so each sequence, each segment or each gene is carrying specific information. Okay, so there are some gene like Z, Y, okay, and each gene is going to code or express the particular protein. So DNA first will form RNA and then from RNA protein will get formed. Okay, so RNA polymerase will come, which is the enzyme, bind to that. Okay, this RNA polymerase will go read the DNA and make the RNA from the DNA. Okay, but when the lactose will come and bind to that, when the lactose will come and bind to that particular enzyme, okay, then what will happen? This additional site is binded by the lactose. Okay, then what will happen? The additional, here one of the gene is regulatory gene which will release R gene, and this R gene will form repressor or another type of protein. Okay, let's not write the lactose here. So this repressor, which is expressed by this R gene, this enzyme will go and bind with that and it can stop the reaction. It can stop the process, right? So if in the medium lactose is present, which is the inhibitor, if the lactose will come and bind to the allosteric site, what will happen? This enzyme will not be able to go and bind with that, or this substance will, protein will not go and bind to this enzyme. So once it will lactose at the allosteric site, what will happen? Now their activity will get modified. Now it will not go and bind with this enzyme. And now this enzyme will be able to perform their activity. So this inhibitor or allosteric is the site, is the additional site in the enzyme to which any of the substance other than the substrate can bind, okay, and can affect the enzymatic activity. Either it will stop the reaction or it will enhance the reaction, but definitely it does not enhance the reaction because the allosteric is not for the substrate. Are you getting me? Only the to the active site substrate will come and bind. So this one, after the binding with the substrate, the enzyme will perform the catalysis process, the reaction, or not the reaction, the enhances the reaction. It will catalyze the reaction. Are you getting me? So whatever thing other than the substrate, it may come and bind to the other site other than the active site, that is allosteric. Clear? So this is the structure of the uh, enzyme. Okay. So this allosteric site and the enzyme with the allosteric site is called as allosteric enzyme. If any of the allosteric site is there in the enzyme, then that particular enzyme is called as allosteric site. This allosteric site is not mandatory that it will present in each and every type of enzyme. No, it is not mandatory. Okay, so there are some example of allosteric enzyme like hexokinase. Hexokinase is an example of allosteric enzyme. Done? Okay. Now let's see another thing related to enzyme. So there are different terms related to enzyme, okay, that we need to know. For example, endoenzyme. Endo means inside. Okay, if enzymes are functional only inside the cell and not outside the cell, then this type of enzyme is called endoenzyme. Exoenzyme, if enzymes catalyzes the reaction outside the cell, okay, then this type of uh, enzyme is called exoenzyme. So all the digestive enzyme, for example, pepsin, trypsin, okay, amylase, these are what? Exoenzyme. Clear? Proenzyme, zymogen. Proenzyme or zymogen, these are the precursor of enzyme or the inactive form of enzyme. For example, trypsinogen, pepsinogen, okay, pepsinogen. They are present but in inactive form or the precursor form. Once they get the suitable environment, it will activate and turns into the active form pepsin. Are you getting me? Isozyme. Isozyme, enzymes having similar action but little difference in their molecular configuration. Okay? Isozyme, the structure will be the, their uh, function will be the same, only the difference will be in their structure, the molecular configuration, molecular structure. Inducible enzyme. Inducible enzyme, when the enzyme will get formed, okay, only by the induction of substrate. If the substrate is available, then only the enzyme will form. Then this type of enzyme is called inducible enzyme. Means enzyme required something which induce or which help or motivate to get formed. Are you getting me? Biodetergent. If the enzymes are used to wash or remove the stain, or if we are using any enzyme as a washing powder, then this type of enzymes are called as biodetergent. For example, pepane. Okay, pepane. We use pepane as a stain cutter. Housekeeping or constitutive enzyme. These enzymes will present all the time in the cell, okay, and that too in a very constant amount. Means whether the environment is suitable or not, the amount will be the constant. Amount will never change. Okay, and these enzymes are very essential for the metabolic activity or for any enzyme or any chemical reactions in the cell. Okay, so these type of enzymes are called as housekeeping cell enzyme. Nomenclature of enzymes or the classification of enzyme. As per their function, different types of enzymes are there, and we have named them according to their action. So there are basically 6 type of enzymes. Okay, 6 types. How many types of enzymes are there? 6. Okay, and as per their action catalysis process, we name them. For example, oxidoreductase. Oxidoreductase means any enzyme who is having ability to uh, oxidize the thing or reduce the reaction, okay, oxidation process as well as reduction process, then this type of enzymes comes under the category oxidoreductase. So oxidoreductase have 2 different types, oxidase and reductase. If any enzyme is having ability to oxidize the thing, okay, helps in the oxidation. What is the oxidation and reduction? What is oxidation means?
Ma'am, oxidation is addition of oxygen and removal of hydrogen, ma'am.
And
And
reduction?
reduction is removal of oxygen and addition of hydrogen.
Very good. If any enzymes have the ability to transfer, have the ability to transfer any chemical group from one substance to the another substance, from one form to the another form, then this type of enzyme is called as transferase, because it is transferring the chemical group from one reactant, and in the final product you will get that same uh, chemical group into the will attach to the other substance which is present as a other reactant in the reaction site. Are you getting me? For example, we are having A plus B giving, suppose, C. Okay, and at this time A was having COH. The product will maybe A plus C plus D. So once the C and D will form, the COH will transfer from A to the C or D. Are you getting me? So this transferring process is catalyzed by the enzyme is called transferase. Example, kinase, hexokinase. Okay, hydrolysis. Hydrolysis, hydrolysis. If any enzyme have the ability to hydrolyze, okay, hydrolyze. Hydrolyze means
Addition of H2O, ma'am.
addition of H2O, right? So if any enzyme who is accelerating the hydrolysis process, or hydrolyzing the substance, for example, if it is hydrolyzing the protein, then this type of enzyme is proteinases. Hydrolyzing the RNA, then ribonucleases. Hydrolyzing DNA means deoxyribonucleases. Hydrolyzing the fat, lipases. So basically here we are not only adding the water molecule, but by adding the water molecule we are breaking, digesting the substance. So by adding the water we are digesting the protein. By adding the water we are digesting the RNA. By adding the water, we are digesting the DNA. By adding the water, we are digesting fat. Clear? Lysis. 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 Yes. Lysis. If you want to remove any substance, okay, suppose you are removing anything which is attached to C, carbon, okay. So carbon is having ability to bind with 4, right? So if you will remove any R or any H from this carbon to make it stabilize, this carbon will form double bond with the adjacent carbon. right? So this type of reaction will be done by the lysis. Isomerases. Isomerization process done by the enzyme is called isomerases. What is isomerization? Rearrangement of atoms. Rearrangement of atoms in a molecule. So structural organization will change. Are you getting me? Molecular structure will change. The molecular number will be the same, but their arrangement will change, right? So this rearrangement of the structure is done by the enzyme isomerases. Ligases or synthetases. If you want to join some molecule, okay, if you want to join some molecule, this joining is done by the specific type of enzyme which is called ligases. Okay. For example, if you want to join two nucleotide, okay, you will use ligases enzyme. Polymerases. If you want to increase the number, okay, polymerization, then you will use the enzyme polymerases. Clear? So mainly there are six. How the six? Because hydrolysis and lysis, okay, hydrolysis and lysis, they are doing their function nearby same, okay. Are you getting me? Normally there are six, okay, but if you will go more specific, you will find seven different types of naming, okay. Nomenclature naming is seven types, okay. Next is enzyme action. So before we move to the enzyme action, let's take a small break and then we will continue. Let's meet after 5 minutes, okay. Chalo. Okay, now let's see the enzyme, okay. When we talk about enzymatic action, the most important part is activation energy. To uh, to make a product or to reach or to convert the reactant into the final product, the reactant required some amount of energy, right? So activation energy is the minimum amount of extra energy which is required by the reactant to perform the reaction and gets converted into a product. And this minimum extra energy is what? Activation of energy. So this energy actually helps to convert the reactant or molecule into the product. So here you can see, suppose this is your reactant. Okay. Okay. Here this is your reactant. So this reactant will take some more extra amount of energy so that the reactant will form product. Okay. When there will be no enzyme, the activation of energy will be high. Why? Why? What is the definition of enzyme, or what is the property of enzyme? Enzyme cannot start chemical reaction. It simply enhances, accelerate the reaction. Okay. When the enzyme is not there, means no supportive system is there to accelerate the reaction. At that time, reactant required more energy. Okay. And that's why you can see the graph of activation of energy is very steep, right? Whereas once you add the enzyme to that, so now the reactant is having enzyme, and enzyme have the ability to accelerate the reaction, right? So here the activation energy will low down. Okay, so we can say that enzyme lowers the activation energy. Okay, because enzyme itself enough to accelerate the reaction. Are you getting me? So there are different types of theories related to the enzymatic action. For example, lock and key theory, and the other theory is enzyme substrate complex theory. Okay. So this theory, lock and key theory, this is also called as template theory, and this was given by the Emil Fischer. As per this theory, the active site of enzyme, what is the enzyme here? The lock. Active site of enzyme, okay, serves as a lock. And here they have a small site to which the key can attach to that. Okay, so here the key is substrate. When this substrate will binds to that particular site in the enzyme, in the lock, okay, they will now what they will do, they will make a complex, lock and key complex. So the same way, the active site of the enzyme when binds with the specific substrate, now they will make a enzyme substrate complex. And once they will form an enzyme substrate complex, they will perform the or accelerate the reaction. Okay. So here the active site is very specific to the specific substrate. Only the particular active site will bind to their particular type of enzyme. So we can say that enzymes are substrate specific. Clear? Other than the particular substrate, if anything else will come and bind to the active site, it will not get bind, because you can see the structure, the shape of the active site and the shape of the substrate is same. So this substrate will only fit to that area. If you are having the substrate like this, it will not fit to this. Okay, and once it will not fit, they will not form a complex. Another theory is enzyme substrate complex theory, and this theory was given by the Michaelis and Menten. This Michaelis and Menten proposed this theory in 1913, and they said for any catalytic reaction to happen, it is necessary that enzyme will bind to the substrate and make a complex. Then only this complex will accelerate the and it will help to make a product. Okay. If enzyme will not bind to the substrate reactant, they will not form the product. Okay. Only in the reaction will get accelerate and it will form product. Clear? Now let's see the another theory related to the enzyme action, that is induced fit theory. This is not actually the theory, but this is the hypothesis because it was not proved. Okay. So this hypothesis, as per this hypothesis, oh, actually this hypothesis was given by the Koshland. So as per this hypothesis, the active site, what is the active site here? This one. This is the active site of enzyme. Okay, so as per this theory, the active site is not static, but it undergoes the process of changes. What type of changes? Conformational changes. Okay, it undergoes the process of conformational changes so that substrate will bind to this active site, which is not correct. Okay, the specific substrate as per their conformation will come and bind to that. The enzyme will not change their conformation as per the substrate. Okay, and that's why this is still a hypothesis. Okay, then we have seen the inducible enzyme, right? Inducible enzyme. For inducible enzyme, yes, we can say that active site induced by some inducible substance so that they can fit with the particular substrate. But this is not generalized theory or hypothesis for each and every type of enzyme. Clear? Now let's see the enzyme action is affected by different factors. There are some factors who can affect the enzyme action, and the first one is pH. How the pH will affect, I think we already discussed, right? How the pH will get affected. How the pH will affect the enzymatic action. As we know that enzymes are very sensitive or
Sensitive.
pH specific, temperature specific, right? So each enzyme shows its highest activity at the optimum pH, that is 7. Okay, the activity will decline in both the condition if the pH is lower than the optimum or above the optimum value. Temperature. Yes, can you explain me temperature? Low temperature will preserve the enzyme for a certain period of time. So it is temporarily the enzyme will become inactive.
Inactive.
Once it will get the optimum temperature, again it will become active. If the temperature is higher than the optimum one, that is 37 degrees Celsius,
37
at
degrees
that time what
Celsius.
will happen? The enzymatic activity will destroy
Teenage.
or the enzyme will denature because of the heat. Heat can denature the protein, right?
Yes, ma'am.
Yes. So you can see the graph. The graph is very important for each and every factor, okay? Another is concentration. When we were talking about the concentration in the property, we have seen the Michaelis-Menten equation, the Km constant, right? Km constant. Yes. What we have seen about the Km constant.
Number of molecule substrate converted into product per unit time, ma'am.
Okay. Chalo, let's see the concentration of enzymes. As we already know that very small amount of enzyme is enough to accelerate any reaction. Okay, that means the rate of reaction is directly proportional to the enzyme concentration. If the enzyme concentration will increase, then what will happen? if the enzyme concentration will increase, if you increase the amount of enzyme, what will happen? It will increase the rate of reaction up to a certain point, after which the rate of reaction becomes constant. Are you getting me? And
ma'am.
when the enzyme concentration is very low, okay, below the normal range, then what will happen? The reaction will not take place, right? So increasing enzyme concentration increases the number of available active site. Please write it. Increase the enzyme concentration. Enzyme concentration increases the number of available active site. So more and more substrate will come and bind to that, and it will increases the rate of reaction. But at certain point, the rate of reaction will be constant.
Constant.
Okay. Now let's see the concentration of substrate. How the concentration of substrate affect the enzymatic action. So increase in the substrate concentration, it will increases the activity of enzyme until all the active sites of enzymes are saturated by the substrate molecule. Okay, so here you can see as the substrate concentration is increasing, the rate of reaction is also increasing. Only till the time when the active site is all the available active site is actually saturated by the substrate concentration, or sorry, available substrate. Okay, once all the active site becomes saturated, now what will happen? The rate of reaction will be constant for a certain period of time, and then now there will be no more available active sites, so no more enzymatic reaction will take place. Clear?
Yes, ma'am.
And that's why the substrate molecule occupy the active site vacated by the product and cannot increase the rate of reaction further. Clear?
Yes, ma'am.
Okay, so to the substrate concentration, we are having a constant, and that is Km constant. Okay, now let's see the Km constant. How the constant, or the turnaround number, or the Michaelis-Menten constant, okay, is related to the enzymatic activity. So Km constant or Michaelis-Menten constant of an enzyme is actually what? Concentration of substrate at which the rate of reaction of the enzyme will attain its half, right? Half of the maximum velocity. So as you can see here, the total rate of reaction is this, and the Km constant, you will find the half of the maximum velocity. Okay, are you getting me? So the value of Km constant should be lower for the enzyme. Okay, half of the rate of reaction. So what will be the value of Km? Half of velocity, maximum velocity. Km is what? Half of maximum velocity of enzymatic action or the reaction. Okay, so this Km constant exhibits the catalytic activity of enzyme, and this Km value will be different. Okay, different from substrate to substrate. Why it will be different from substrate to substrate? Because different enzyme is having different affinity towards the different substrate, right? All the enzymes do not have a same affinity for the different substrate. Are you getting me? So different enzymes having different affinity for towards the different substrate, and that's why Km value will always vary. Okay, higher the Km value, if the Km is higher, more, then if the Km is high, which means a higher Km indicates low affinity. Why? Whereas lower Km shows the strong affinity. Here the affinity means the foundation or the complex forming affinity between the enzyme and substrate. Are you getting me?
Yes, ma'am.
So if the Km is high, which means the affinity between the substrate and the active site, okay, is low. If the Km is low, which means substrate and active site, the affinity between the active site and substrate is high. Okay, so as you can see, this E high means enzymatic activity. This E low means enzymatic activity. Okay, are you getting me?
Yes, ma'am. Affinity means bond between substrate and
Ability to bind with the substrate.
yes, ma'am.
Clear?
Yes, ma'am.
So let's take an example of protease. Protease is what? A type of enzyme, right? So protease acts on the protein. It will digest the protein, right? So there are various types of protein. There are not only one type of protein. Even thus, as per the structural organization, as per the uh, addition of alkyl group in that, branching, unbranching, right? There are various types of protein. So this protease will not act on each and every type of protein, right? And that's why the Km value will be different from protein to protein, though the enzyme is same. Are you getting me?
Yes, ma'am.
And hence, Michaelis-Menten equation or Km value describes how the rate of reaction relatively varies with the substrate concentration. So can we say that oh, V naught, this one, V naught is equal to V max of the substrate divided by Km constant plus substrate. Okay, here the V naught means the rate of initial reaction. Okay, the rate of initial reaction from where the reaction is initiating, the rate of initial reaction. Okay, V max means maximum relative or the reaction rate at which the maximum substrate is actually make a enzyme substrate complex. Are you getting me? And
Yes,
Km, you
ma'am.
already know, this is the Michaelis-Menten equation. Okay, so
Yes, ma'am.
the K2 plus K3, anything is what? Your substrate concentration. Okay, so this is all about the Michaelis-Menten equation. So this equation actually shows the affinity. Okay, the greater the affinity between the enzyme and substrate, lower the Km or the Michaelis-Menten equation. Okay,
Yes, ma'am.
now let's see the question. Which one of the following statement is incorrect? A competitive inhibitor reacts reversibly with the enzyme to form enzyme inhibitor. A competitive inhibitor In the competitive inhibitor, the inhibitor molecule is not chemically changed by the enzyme. The competitive inhibitor does not affect the rate of breakdown of enzyme substrate molecule. The presence of competitive inhibitor decreases the Km of the enzyme of substrate. Nothing is going inside the head. Yeah, right?
Ma'am?
Hmm. I think this is completely different topic, ma'am. Right? Huh?
Yes.
While we were discussing about the allosteric enzyme, at that time I told you that there are something which when it will be attached to the allosteric and it can inhibit the enzymatic action, right? Right? So here we are going to first we need to know about the inhibition of enzymatic action. Okay? Inhibition of enzymatic action. So enzyme can be or enzymatic action can be inhibited by the denaturation process, that is by the adverse temperature or pH. So first is denaturation of enzyme, okay, or the protein. Denaturation. This is the one way to inhibit the enzymatic action, okay. That is I'm not going to explain because you already know, right?
Yes, ma'am.
Another way to inhibit the enzymatic action is by competitive inhibition. Competitive inhibition. What is the meaning of inhibition? To stop, right? Competitive means something is competing with the substrate, okay? And if that something, that substance will win this race, okay, binds with the allosteric site, binds with the protein part of the enzyme, and it will inhibit the enzymatic reaction. For example, the malonate, okay, malonate or the malonic acid. Malonic acid. This malonic acid is also known as malonate. Okay, this malonic acid will compete with the succinic acid. Why? Actually, what happens when sometimes the substrate, two different substrate is having a similar type of configuration. Okay, if any substrate is having a similar type of configuration, then there will be the competition. This is quite normal, right? Right? So they start competing, and whoever will win and binds with the active site of the enzyme, it will change the reaction completely, right? For example, the actual reaction is what? The succinic acid need to dehydrogenate and make a fumaric acid, right? But the chemical configuration of succinic acid is similar to the malonic acid or succinate. The chemical configuration of succinate is similar to the malonate. Succinic acid or the succinate. Okay, they have the similar configuration. Are you getting me,
Yes,
Aarti?
ma'am. Yes, ma'am.
So actual reaction should be succinate, okay, in the presence of succinate dehydrogenase will form fumaric acid, or succinic acid will form fumaric acid in the presence of which enzyme? Succinate. You will write the full form of S, okay?
Yes,
Succinate.
ma'am.
Succinate dehydrogenase. Okay, so in the presence of succinate dehydrogenase, this succinate will transform and converted into the fumaric acid. But this succinate and this malonate, they both have the similar type of configuration. So what will happen? Instead of succinate, this malonate will come and bind to the active site of the protein. Okay,
Yes,
and
ma'am.
because of that, what will happen? This enzyme will not work properly. Okay, so the reaction will not take place. Are you getting me? So competition will happen because of the chemical, similar type of chemical configuration. Clear?
Yes, ma'am.
Another way to inhibit the chemical reaction, enzymatic action by the non-competitive inhibition. This is the third. Non-competitive inhibition. Okay, so here competition is not taking place, right? The different types of substrate, okay, or the similar type of substrate, similar type of different substrate, they are not competing with each other for the active site, right? Right? But
Yes,
what
ma'am.
will happen? They are not competing, but the similar type of or other chemical will come and bind to the other site. Other site means the site other than the active site. Okay, and by binding or attaching with the other side, the allosteric site, okay, they are changing the configuration. Okay, they are changing the configuration. So here it is not specific that always they will bind with the allosteric enzyme. They will bind with the enzyme at the site, but other than the active site. Are you getting me? It may be the allosteric also. Okay, so because of this bonding, what will happen? The configuration will change, and once the configuration will change, what will happen? The enzyme will not work properly. Okay, either these substance will change the configuration of active site, or will it will make the shape of the enzyme into the different. They will change the configuration of whole enzyme structure. Are you getting me? So this is
Yes,
what
ma'am.
non-competitive inhibition. Clear? The
Yes,
chemicals
ma'am.
are not binding with the active site but the other site, okay, and changing the configuration of active site so that this particular protein will not bind with that, with the particular substrate, and once they will not bind with the particular substrate, enzymatic action will not take place, and that's how they are inhibiting the enzymatic action. Clear?
Yes, ma'am.
Another way is the allosteric inhibition. The fourth one is allosteric. Here it is not mandatory. Okay, if any enzymatic action is getting inhibited by adding or attaching with the allosteric, then it will comes under the allosteric inhibition. Okay, if any substrate or any chemical is binding with the enzyme or any site in the enzyme other than the active, it will comes under the non-competitive. Okay, so let's see the allosteric inhibition. This allosteric inhibition is also called as feedback inhibition. So this feedback inhibition or allosteric inhibition, before we move to that, first we need to know what is allosteric enzyme. If the enzyme is having allosteric site along with the active site, right? So the chemicals will bind to this allosteric site and inhibit the chemical reaction. If the inhibitor is binding to that site, then they will stop the reaction. Let's take an example. For example, glucose during the process of glycolysis. Okay, glucose. Glucose in the presence of ATP, okay, it will form glucose 6 phosphate. Glucose 6 phosphate. Okay, plus as it is ATP, so it will be ADP because one of the phosphate will come and bind to this glucose. So it will become glucose 6 phosphate. Clear? Yes. And this reaction will take place in the presence of enzyme hexokinase. Kinase means kinase. Hexokinase. What is the function of kinases? Mm. Yes, RT.
Transfer,
Kinase.
ma'am.
Sorry,
Transfer.
kinase will transfer. Sure. Yes. So here, one of the molecule, this enzyme is transferring from here to here. So it is glucose 6 phosphate. Okay. Once the glucose 6 phosphate will form, this glucose 6 phosphate will come and bind to the allosteric site of hexokinase. So it will come and bind to the hexokinase, where at the allosteric site. Once the glucose 6 phosphate will binds to the allosteric site of hexokinase, now the hexokinase will stop functioning. So this is the allosteric inhibition. Okay, clear? This is also called as feedback inhibition. Once you will start uh, going through the next chapter, photosynthesis, all the reaction you will study in that chapter, photosynthesis, and that is, I think, next to next chapter. So this is all about the enzymatic action. Any doubt? Now let's see the question. Yes. So now the question is, which of the following statement is incorrect? Competitive inhibitor reacts reversibly with the enzyme to form an enzyme inhibitor. No. Right? In competitive inhibition, inhibitor molecule is not chemically changed by the enzyme. Yes. Is it so? Answer it quickly. Yes, answer it quickly. See, just now we have discussed now.
Yes, ma'am. Incorrect only they asking, ma'am.
Yeah, incorrect. Chalo, the competitive, actually this is correct. Okay. The competitive inhibitor does not affect the rate of breakdown of enzyme substrate complex. competitive inhibitor, the malonate. Malonate does not affect the rate of breakdown of enzyme substrate complex. Yes, it does not affect, right? Right? So again, this is the correct. The presence of competitive inhibitor decreases the km of the enzyme of the substrate because they are competing for the competing with the substrate for the active site, right? And Km constant is related toward the substrate concentration, right? And that's why the presence of competitive inhibitor will not decrease the Km of the enzyme of the substrate. So what will be the answer? The incorrect one is option
D, ma'am.
yes. Okay.
Yes, ma'am.
This competitive inhibitor is what? The competitor is the structurally or similar to their configuration with the other one, other substrate. And that's why they are competing for the active site, right? So in the
Yes, ma'am.
presence of that, the competitive inhibitor, okay, in the presence of competitive inhibitor, in the presence of malonic acid, what will happen? It will decreases the affinity of substrate to bind with the active site. And once the affinity will decrease, automatically the Km constant will increase because they are inversely proportional, right?
Yes, ma'am.
Feedback inhibition is caused by blocking of enzyme by accumulated end product, chemical produced by hormones competitive inhibition. In feedback inhibition, hmm, glucose 6-phosphate, what was that? Accumulated end product.
Products.
Okay, so this end product will come and bind to the enzyme, and that's how they will stop the reaction, right? Enzymatic action. So what will be the answer?
A.
A. So this is all about the enzyme, the structure, the properties, and mechanism or the action, enzyme action. Clear?
Yes, ma'am.
Akka?
Ma'am, what is the major difference in non-competitive inhibition and the allosteric inhibition, ma'am?
Okay. So here, where is the structure? Okay. This is your allosteric site. Okay.
Yes, ma'am.
In allosteric inhibition, the product and product will come, okay, after the enzyme substrate complex, the reaction will take place and product will form, okay, and continuously this process is going on and the product is getting accumulated, depositing at that place. Now once the product concentration will increase, this product will come and bind to this allosteric site. Once it will bind to the allosteric site, it will change the affinity. Are you getting me?
Yes,
And
ma'am.
now this enzyme will no more able to bind with the substrate and perform the activity. This is the allosteric inhibition, right? Non-competitive inhibition means the substrate, not the product, okay? The substrate, the similar type of substrate will bind to the enzyme other than the active site. They are not saying that allosteric, any site, but other than the active site. It may be bind this site, anywhere. And it will change the configuration of the active site. It will change this shape, configuration. Okay, so now this active site is no more available to bind with the particular substrate to perform the action. Are you getting me?
Yes, ma'am.
So this is what? Non-competitive. Clear?
Yes, ma'am.
Okay. Anything else?
No, ma'am.
Pakka. See,
Yes,
this
ma'am.
enzyme part is very important and very confusing also. Okay, here I am going to give you one more classification chart for enzyme nomenclature. Okay, so you can refer either of them. Okay, this one is more elaborative because here the class enzyme is classified on the basis of different criteria. So here the enzyme is classified on the basis of substrate, on the basis of type of reaction. Okay, as per the substrate, there are three types: protease for protein, carbohydrases for carbohydrate, lipases for lipid. Okay, and on the basis of type of reaction, if oxidation and reduction is taking place, so it is oxidoreductase. If transfer of molecule is taking place, so transferase. If hydrolysis or the breaking down of the any molecule by the addition of water, then it will be hydrolysis. If breaking down of any of the substances by adding the water, lysis, isomerasis, and ligases. Okay, here polymerase is missing. Okay, are you getting me? So these two are little bit similar, and the additional one is polymerase. Okay, so polymerase is the new one. Okay, but actually there are 6 only. Clear? So you can
Yes,
refer
ma'am.
any of the chart. Okay, Aarti, please go and check your dashboard. Are you able to see your class assessment or not?
Ma'am, can I refresh and come, ma'am?
Yes, please. Are you able to see, Aarti? If
Yes,
yes, then
ma'am.
do like this.
Yes, ma'am.
Okay, chalo. Please start submitting your answer. Suppose you will not be able to finish your assessment by 9:30. So don't worry. Next class is our DRS. At that time, first we will discuss the question. Then I will solve your query because I know this chapter is very confusing. And then I will start shooting my doubt.